Welcome again, class. This is Dr. Collins, and we're going to continue our discussions on immunity, chapter 2, and this is going to focus on immune receptors and phagocytosis. So let's continue. Immune receptors and phagocytosis. So what happens first? Of course, we need recognition. And we have Nobel Prize winners again here, and they were awarded for their discoveries concerning the activation of innate immunity. So basically what happened is that they were able to locate the specific receptors, cell receptors that will initiate immunity in our special immune cells. So these receptors are called toll-like receptors, and they are located both on the cell membranes so that they can detect external pathogens, and also internally so they can catch those pathogens that work their way inside the cell. Viruses are a good example. These are a type of pattern recognition receptor, and these are only found on phagocytic immune cells. And they are the primary sensors that, when activated, will initiate and moderate the cell's immune responses. So what happens? Well, these toll-like receptors will bind only to very specific molecular patterns, and they break down into 3 different types. We have PAMPs, DAMPs, and SAMPs. So I want you to just notice right away the first 2, PAMPs and DAMPs, are actually the ones that promote the inflammatory response, whereas SAMPs counteract that. And you can see it in the name, suppressor-associated molecular patterns. We're going to talk about these more in detail next. So PAMPs. These are unique molecular structures that are only found on pathogens. So examples would be bacterial cell walls, flagella, viral capsids. So these are basically the capsules that surround the viral DNA and RNA and allow the viruses to escape from the cells where they reproduce and then move on and infect other cells. So the important thing about PAMPs is they distinguish the self from the non-self. And this is a very important concept in immunity. So we have lots of immune cells, but we only want those cells to attack invaders that come into us that are not our own cells. So this is a very important concept, and we'll be talking about this a lot more in our next lecture. When the receptors bind, this triggers 2 main events. 1, phagocytosis, and 2, the release and synthesis of various inflammatory cytokines and also interferons. So I have mentioned interferons before. They are involved in preventing the spread of viral infections. And this is just a picture that shows you what kind of happens here. We have the pathogen. Let's say that's a bacteria. We have the receptor, the toll-like receptor. And if you look closely, you can see that the pattern on the bacteria in this case and on the receptor are identical. In other words, it's like a lock fitting into a key. And this is important for the recognition process. These receptors will only interact with one specific type of molecular pattern. Now, DAMPs are similar but very different. So DAMPs are actually molecules that come from our own cells. And they are molecules that signal danger or damage is occurring in the body. So these particular molecules are recognized by the macrophages, and it will stimulate them to release pro-inflammatory cytokines. So basically you have a sterile inflammation. The cause of the problem is not the actual pathogen, but the results of the pathogen infecting the body. So you will get danger signals being sent from from cells that are initially infected with something. Damage signals come more from the cells that have actually undergone injury or are dying from necrosis, and they are releasing their cellular contents into the environment around them. So these are recognized as something that the macrophages respond to. If the DAMP release is excessive or prolonged, then the macrophages are going to keep responding and over-responding, and then you will get tissue damage occurring. And this is what happens in COVID. And I'll be talking about that a little bit later here. This is just an example, a picture of DAMP release. You can see the cell. It's been subjected to severe injury. It's basically undergoing necrosis now. The membrane's breaking down and it's releasing its contents. And some of these will be identified as probably danger and damage molecules and will stimulate the macrophages to respond. So cytokine storm, I've mentioned this before, I believe, but let me explain it a little bit further. So again, we're talking about inflammation and what is causing the inflammation and where this leads to. So with the PAMPs, they are only related to the actual pathogens that are involved. And we can see a whole bunch of them here in this portion of the slide. And if you follow that through, they present with PAMPs to the toll-like receptors, and then the toll-like receptors will get activated, and then the immune cells will do both cytokine release and actually phagocytosis to get rid of these pathogens. However, these also cause cell injury and tissue injury. You see that also with things that aren't pathogens. So in trauma, burn, pancreatitis, surgery, all of these conditions and many others will cause this injury to the cells, and that will then result in the release of the DAMPs. So again, these DAMPs will stimulate the toll-like receptors, and in this case, you're going to get a release of cytokines and often a massive release, which will cause this cytokine storm. So when this happens, you're getting more tissue damage, more DAMPs are released, then more macrophages respond, and you can see how this is a vicious cycle. So there has to be something that's going to prevent this and control the inflammatory response, and that is where our SAMPs come together. So remember, this is a suppressive associated molecular patterns. So these are molecules that will suppress the immune responses. Only very few have been identified, and there's a lot of research going on in this field to identify more. And it's the interaction between the PAMPs and the DAMPs, and then the SAMPs, which will control the inflammatory response. The problem is if this control is lost and the system becomes dysregulated. If you have a situation where the PAMPs and DAMPs predominate, you're going to have extensive tissue damage, and that will eventually lead to multiple organ failure in the body, and that leads to death. If the SAMPs predominate, so now we're having more a suppressor effect, you do get immunosuppression. So basically what happens is the immune cells aren't responding as much as they should, and you're not having as much occurring to get rid of whatever is causing the problem. So with that immunosuppression, you will get pathogens that are escaping the process of being eradicated, and you will get increased infections. And depending on what the infection is and how severe, and if it's responsive to medications, this also can lead to death. So this chart shows a little bit about some of the interactions here between DAMPs and PAMPs. And I like to talk about COVID using this chart because I think it'll explain a lot of what happens with the disease, the COVID disease. So obviously it's caused by a virus. The virus is actually the initial stage of COVID. After the virus infects the cells, after about a week or so, pretty much all of the virus is eradicated. And if you continue to have sickness or illness, then you're dealing with the cytokine problem. You have excess cytokine, and you can develop a cytokine storm situation. And then the next step would be that this is so destructive to the body that you end up with that organ failure that will occur. So if we look here at the damp portion, anything you see under this dotted line is what is controlled inflammation. Anything above is where you have excess occurring. And so initially with COVID, you'll have this controlled, and then it starts to get over into this situation where it's no longer controlled, it's out of control, and you get the eventually the death if you can't treat that. And then underneath here, you have the SAMP levels. This is, again, in levels above the dotted line. That is a controlled situation. As you get outside of this line, you start getting into more and more problems with the immune system not responding anymore as it should. It's being suppressed. And then as you get more and more of this occurring, you end up with this susceptibility of infection. So what can happen next? Well, of course, we're going to talk about phagocytosis. And this is where the pathogen is engulfed and killed and eventually degraded. So a few basic facts about phagocytosis. So interestingly enough, almost all of our cells can phagocytize, but generally not very well. We have our specialized immune cells that are the best at it. And the neutrophils are the most efficient, followed by macrophages and then the dendritic cells. So this is the method that the cells are able to directly kill and eliminate pathogens. And it is a process that is enhanced by opsinin. Remember, we talked about that with complement, that some of those complement proteins can actually coat the pathogens and so make them become very recognizable by the phagocytes, much more so than otherwise, and aid certainly in the phagocytic process. Also, the process is enhanced by cytokine stimulation, which actually is kind of like an extra boost to the the phagocytes, so that they become even more effective and aggressive in going after the pathogens. Now, the important thing, very important thing that phagocytosis also does, because it breaks down the pathogen into little particles, those little particles actually are used to activate the acquired immune system. That's a system we're going to talk about next week. So if you don't have phagocytosis occurring, you're actually not going to be able to have an immune system that's going to keep you alive. You absolutely need to have that happen in order to completely fight infections. So we'll use an example of bacteria. So after the immune cell binds to that bacteria with their toll-like receptors, the membranes of the cell and the bacteria wall will attach. And also a variety of co-receptors will also bind to the bacteria, and then this strengthens the attachment. And this is what you need. You have these wiggly bacteria, for example. You need to be able to really hold on to them very firmly. After this occurs, we have engulfment. The pathogen is brought into the cell, and we have a phagosome that is formed. The phagosomes that then fuses with lysosomes, and you get a phagolysosome. So if this sounds familiar, it is. It's very similar to the process of autophagy, but autophagy deals not with these pathogens that have come in. In fact, it deals with the existing cell cytoplasm and kind of getting rid of that. So in the process, we have that phagolysosome, And then we have the bactericidal substances released and enzymes, various enzymes that will kill and digest the bacteria. So we have a chart here. This just shows you a picture, I should say. This is a neutrophil. It has the multi-lobes. Also the granules needed, the lysosome and other granules needed to kill the bacteria. Here you see the binding of the receptors to the bacteria. Here the bacteria is being pulled in and you're getting some oxygen radicals released. We see hydrogen peroxide here and some other oxygen containing molecules. Eventually, of course, the bacteria will be completely engulfed. We have the, in this case, the phagolysosome created. Here you have the contents being released from the lysosomes, and then you have that bacteria eventually killed and destroyed and digested. This is a good video. I recommend you watch this. It is in your modules, and it will show you the actual process of this occurring under a microscope. It's very interesting. So finally, at the very end, we have those digested fragments from the bacteria. Most of these are expelled as waste as a process called exostosis. And also you have those bits that are released into the cytoplasm to be reused by the cell. And as I mentioned before, this is used to initiate acquired immunity. It will eventually activate the lymphocytes, which have been dormant until now, and also stimulate antibody production. Bad news is that some pathogens do manage to evade these processes. They have developed defenses against phagocytosis. So these include a resistant cell wall and membranes, release of various chemical substances and or avoidance of the phagocyte or even the process. So what can happen? Well, one is these pathogens can release things that will inhibit the engulfment. Sometimes the pathogens just work their cells inside the host cells and kind of hide and go dormant until they reactivate at another time. Pathogens can survive inside the phagocytes. Some are able to inhibit the fusion of the lysosome to the phagosome, and others are able to inhibit the effectiveness of the lysosomal contents. So they secrete something that counteracts all of those enzymes and chemicals that would normally harm them. Other pathogens are able to escape from the phagosomes, and some of them just directly kill or damage the phagocyte before or after ingestion. So these types of pathogens are actually resistant to our immune responses and can be very difficult to kill. Some can't be eradicated without very strong medications. For some, we don't even have medications really, and it's just a question of dealing with the symptoms and hopefully making the patient through the particular infection. So examples of the diseases that these pathogens can cause are anthrax, pneumonia, tuberculosis, leprosy, plague, and Ebola. And you've probably all heard about the Ebola virus. There are outbreaks that periodically occur in Africa. This is a very lethal virus. I think we're up to over 50% of deaths occur from it. And unfortunately, it has been isolated, and there's various experiments being done on these viruses. So hopefully this will not escape as one of these laboratories the way the coronavirus did, because a outbreak of Ebola is much more lethal and serious than what we've been seeing with the coronavirus. And that is the end. So please study this material. You will be tested on it. If you have any questions, get back in touch with me, and I will see you in class.